Hello and welcome everyone. I'm Dr. Muzahid and this is Modern Microbiology Academy. Today we're diving into innate nonspecific host defenses. This is the immune system's rapid response team, the defense that acts before the body even knows what the invader is. If you're studying cardiotechnology, this topic matters a lot because the bloodstream and cardiovascular system are prime targets for infection. Understanding how the body naturally defends itself will help you understand how infections affect the heart and blood vessels and how medical interventions support these defenses. So let's get started. Here's our roadmap for today. We'll start with physical defenses, the body's walls and gates. Then we'll explore chemical defenses, the biochemical arsenal your body deploys. Next, we'll meet the cellular defenses, the white blood cells that act as soldiers and cleanup crews. After that, we'll see how these cells recognize and engulf pathogens in a process called phagocytosis. Finally, we'll discuss inflammation and fever, the body's emergency broadcast system. By the end, you'll see how these layers work together to protect you and how they relate directly to cardiovascular care. Let's dive into the first layer. Before we get into the details, let's clarify what innate immunity really means. Unlike adaptive immunity, which is specific, slow, and has memory, innate immunity is non-specific, rapid, and has no memory. It's like having a security system that reacts the same way to any intruder, whether it's a bacterium, a virus, or a fungus. It's your body's first line of defense and it's always on duty. For example, when you get a small cut, your skin and immune cells jump into action immediately, long before your body produces specific antibodies. This is especially important in cardiology because bloodstream infections can quickly reach the heart, causing conditions like endocarditis. So, a strong innate system is your first shield against cardiovascular infections. Let's start with physical defenses. Think of these as your body's architecture and landscaping. There are three main parts. First, physical barriers like skin and mucous membranes that block microbes from entering. Second, mechanical defenses, coughing and skin shedding that remove pathogens. Third, the microbiome, friendly bacteria that compete with invaders. These defenses work together. Your skin isn't just a wall, it constantly renews itself, carrying microbes away. And your gut bacteria act like friendly neighbors, keeping troublemakers out. In cardiology, breaks in these barriers, like intravenous lines or surgical incisions, can let infections reach the heart. So, intact physical defenses are critical. Now, let's zoom in on your skin, the largest organ and a powerful barrier. It has three layers. On top is the epidermis, made of dead cells packed with keratin, which bacteria struggle to digest. Below that is the dermis, containing sweat glands, hair follicles, and blood vessels. Beneath that is the hypodermis, a fatty layer with more vessels. Your skin protects you in two key ways. First, its acidic pH inhibits bacterial growth. Second, it sheds constantly, carrying microbes away. A small, clean cut heals quickly, but a deep wound can allow bacteria like Staphylococcus to enter the bloodstream. For cardiac patients, skin integrity is critical because infections can lead to sepsis or endocarditis. Next, let's talk about mucous membranes. These line the respiratory, digestive, urinary, and reproductive tracts where the body meets the outside world. They're made of epithelial cells with tight junctions and they secrete mucus, a sticky fluid that traps microbes. In the respiratory tract, tiny hair-like structures called cilia beat together, moving mucus upward, the mucociliary escalator. Coughing and sneezing help expel it. Smoking damages cilia, which is why smokers get more infections. In cardiology, think about ventilated patients. When the mucociliary escalator is impaired, the risk of ventilator associated pneumonia increases, placing extra strain on the heart. Now, mechanical defenses are all about movement. Your body is constantly in motion to remove pathogens. You shed skin cells, you cough. Your intestines perform peristalsis, wave like contractions that push microbes out. Urine flushes the urinary tract. Tears wash the eyes. Even blinking removes debris. But what happens when movement is disrupted? A urinary catheter, for example, bypasses urine flow, allowing bacteria to climb upward. This can lead to urosepsis, which may strain the heart. That's why in clinical care we minimize invasive devices to protect these natural defenses. Let's not forget the microbiome, the trillions of microbes that live on and inside you. These aren't parasites, they're protective partners. They compete with pathogens for space and nutrients, crowding out the bad guys. Some even produce antimicrobial substances like bacteriocins. But when this balance is disrupted, often by broad spectrum antibiotics, we get dysbiosis. A classic example is Clostridium difficile infection, which can be life threatening. Sometimes treatment involves a fecal transplant, restoring healthy microbes. In cardiology, we now know gut health influences inflammation and even atherosclerosis. A healthy microbiome supports a healthy heart. Moving from physical to molecular defenses, let's explore chemical defenses. Your body produces a wide array of chemical weapons that attack pathogens. These include everyday fluids like sebum and stomach acid, as well as specialized molecules like antimicrobial peptides, complement proteins, and cytokines. Each has a unique role. Some break down bacterial cell walls, some poke holes in membranes, some act as alarm signals. In cardiology, we measure some of these chemicals, like C reactive protein, CRP, as markers of inflammation and cardiovascular risk. These aren't just concepts, they're measurable indicators of health and disease. Let's zoom in on antimicrobial peptides or AMPs, small but powerful proteins that act like molecular daggers against microbes. They're produced by your cells and your microbiome. Defensins made by skin and immune cells punch holes in bacterial membranes. Cathelicidin in your skin fights bacteria and fungi. Bacteriocins from gut bacteria keep other bacteria in check. Dermcidin in sweat and histatins in saliva target fungi. AMPs are nonspecific. They attack microbes based on general features, not specific antigens. In cardiology, researchers are exploring AMPs as potential new antibiotics, especially for drug resistant infections that can affect heart valves. Now, meet the complement system, a group of over 30 proteins in your blood that work together like a biochemical cascade. It can be activated in three ways. First, the classical pathway needs antibodies linking innate and adaptive immunity. Second, the alternative pathway is spontaneous, always ready to go. Third, the lectin pathway is triggered when mannose binding lectin binds sugars on microbial surfaces. Once activated, complement proteins do four main things: opsonization, coating bacteria so phagocytes can eat them more easily; chemotaxis, attracting immune cells to the infection site; inflammation, increasing blood flow and permeability; cytolysis, forming a membrane attack complex that pokes holes in gram-negative bacteria. In cardiology, complement activation is a double-edged sword. It helps clear infections but can also damage heart tissue in conditions like rheumatic fever. Cytokines are the messenger molecules of your immune system. They're proteins that cells use to talk to each other. Interleukins coordinate immune responses. Chemokines act as chemical homing signals, guiding white blood cells to infections. Interferons warn nearby cells about viral invasions. Then we have inflammation mediators. Histamine, released by mast cells, causes swelling and redness. Leukotrienes are stronger and longer lasting. Prostaglandins trigger fever, and bradykinin makes blood vessels leaky, leading to edema. In cardiology, we use anti-inflammatory drugs like aspirin, which inhibits prostaglandins to reduce inflammation in coronary arteries. But too much suppression can impair immunity. It's a delicate balance. Now let's meet the cellular defenders, your white blood cells. All blood cells originate from hematopoietic stem cells in the bone marrow through a process called hematopoiesis. These stem cells differentiate into two main lines. The myeloid lineage produces granulocytes, monocytes, and platelets. The lymphoid lineage produces lymphocytes, including natural killer cells, B cells, and T cells. It's like a cellular factory constantly producing new soldiers for your immune army. In cardiology, we monitor white blood cell counts as a sign of infection or inflammation. For example, a high neutrophil count often indicates a bacterial infection like bacterial endocarditis that could affect the heart. Let's look at granulocytes, white blood cells with visible granules in their cytoplasm. First, neutrophils, also called polymorphonuclear leukocytes or PMNS, are your first responders to bacterial infections. They engulf bacteria, release toxic granules, and can form NETs neutrophil extracellular traps, webs of DNA and proteins that catch pathogens. When neutrophils die in battle, they form pus. Next, eosinophils specialize in fighting parasitic worms and participate in allergic reactions. Their granules contain major basic protein, which damages parasite membranes. Finally, basophils release histamine and other inflammatory chemicals. They are like alarm sounders. In cardiology, high eosinophil counts can indicate parasitic infections or allergic reactions that sometimes affect the heart, like eosinophilic myocarditis. Now, let's talk about agranulocytes, white blood cells without visible granules but equally important. First, natural killer NK cells patrol for abnormal cells like virus infected or cancerous cells. They don't need prior exposure. They recognize missing self markers and release perforin and granzymes to trigger apoptosis. Next, monocytes are the largest white blood cells in your bloodstream. When they enter tissues, they mature into macrophages, professional phagocytes that also act as antigen presenting cells, bridging innate and adaptive immunity. In different tissues, they have special names: Kupffer cells in the liver, alveolar macrophages in the lungs. In cardiology, macrophages play a key role in atherosclerosis. They ingest cholesterol, become foam cells, and contribute to plaque buildup. Now, how do these cells find and destroy pathogens? It starts with extravasation. White blood cells squeezing out of blood vessels into infected tissues, guided by chemical signals. Once there, they use pattern recognition receptors or PRRS like toll-like receptors to detect pathogen-associated molecular patterns, structures common to microbes like bacterial cell walls. This binding activates the phagocyte. Sometimes pathogens are opsonized, coated with complement proteins or antibodies, making them easier to recognize. Then, the phagocyte engulfs the pathogen in a process called phagocytosis. In cardiac patients, impaired phagocytosis can lead to persistent infections. For example, in chronic granulomatous disease, neutrophils can't produce reactive oxygen species to kill bacteria. Let's walk through phagocytosis step by step. First, the phagocyte extends pseudopods, arm like projections that surround the pathogen, forming a phagosome. This vesicle fuses with a lysosome full of digestive enzymes, creating a phagolysosome. Inside, the pathogen is broken down by enzymes like lysozyme and by reactive oxygen species produced during the respiratory burst, a surge of oxygen aimed at killing microbes. Finally, leftover debris is expelled via exocytosis, but some pathogens, like the parasite Leishmania, can survive inside phagosomes. In cardiology, we see similar evasion strategies in infections like tuberculosis, which can spread to the heart. Now, let's talk about inflammation, the body's emergency response. Acute inflammation is immediate and localized. Think of a sprained ankle, red, hot, swollen, painful, and hard to use. These signs result from increased blood flow and leaky vessels, allowing immune cells to enter tissues. Chemical mediators like histamine and bradykinin drive this process. But if inflammation doesn't resolve, it becomes chronic. This can lead to granulomas, walled off pockets of infection, as seen in tuberculosis, or tissue scarring, like liver cirrhosis. In cardiology, chronic inflammation is a key driver of atherosclerosis. Inflamed arteries build up plaque, which can rupture, causing heart attacks. So, inflammation is both protective and potentially harmful. Finally, let's talk about fever, a system wide rise in body temperature. It's orchestrated by the hypothalamus, your body's thermostat. Pyrogens, fever inducing molecules, reset this thermostat. Exogenous pyrogens come from pathogens, like LPS from gram negative bacteria. Endogenous pyrogens are made by your own cells, like interleukin 1 and TNF. Fever helps fight infection by inhibiting pathogen growth, since many microbes prefer normal temperature, boosting immune cell activity, and stimulating the liver to release iron sequestering proteins, starving bacteria of iron. But too high a fever can be dangerous, causing dehydration and organ stress. In cardiology, fevers increase heart rate and cardiac output, which can be risky for patients with heart failure. So we monitor fevers closely. Let's tie this all together with a clinical case. Remember Angela from the textbook? She had sudden airway swelling, low complement C4 levels, and a family history of similar attacks. She was diagnosed with hereditary angioedema, a genetic disorder where the C1 inhibitor protein is deficient. Without this inhibitor, the complement system runs unchecked, producing too many anaphylatoxins like C3A and C5A, leading to severe swelling. Treatment involves replacing the C1 inhibitor or using drugs that block these inflammatory pathways. In cardiology, this case shows how complement imbalances can cause vascular leakage and edema. In severe cases, this can reduce cardiac output and destabilize blood pressure, a clear example of innate immunity gone awry. To summarize, innate immunity is a multi-layered rapid response system that protects you around the clock. It consists of physical barriers like your skin and mucous membranes, chemical weapons such as antimicrobial peptides and complement proteins, cellular defenders including neutrophils, macrophages, and NK cells that patrol tissues, recognize pathogens via pattern recognition receptors PRRs, and destroy them via phagocytosis. Systemic responses, inflammation and fever that amplify the defense when needed. For cardiotechnology students, remember these defenses are crucial for preventing cardiovascular infections like endocarditis. Furthermore, imbalances in innate immunity can contribute to chronic diseases like atherosclerosis. Innate immunity isn't just a background system, it's active, dynamic, and intimately linked to heart health. If you found this lecture helpful, please like and subscribe to Modern Microbiology Academy for more in-depth microbiology content tailored for health science students. Want the detailed lecture notes? Just comment lecture notes below and I'll send you a PDF. Have questions or topics you'd like covered? Drop them in the comments. I read everyone. Next time, we'll explore adaptive specific immunity, the smart, memory-based arm of your immune system. Until then, stay curious and keep learning. Thank you for watching.